Assalamu alaikum wa rahmatullahi wa barakatuh. Today I will be talking about the secretion and the absorption of the ions that takes place inside the small intestine. So let's start with the secretions first. Now, before I start, I want to make one thing clear, that is that the secretions actually happen in the intestinal crypt. So all of the water and the electrolytes that actually are secreted in the intestinal crypts over here, and they flow all the way up to the villi and they get absorbed back into the villi. So that's what happens in the crypt. You have secretion, and the villi, as we'll talk about later on, you have absorption. And you might also find it weird. You'll ask this question of why are we actually secreting fluid and electrolytes into the intestinal crypts or into the lumen of the intestine? The answer to that is actually perfectly logical. It is because we need these electrolytes. We need electrolytes like sodium to be able to absorb the amino acid and glucose later on. So we're using the sodium as a means for us to be able to absorb the amino acid and the glucose back into the blood. Okay, we're absorbing the sodium back, but the amino acid and the glucose was part of the chyme that came from the stomach. Okay. So just some FYI information here. The amount of sodium that we have in the chyme or in the diet is actually not enough for us to be able to absorb the content of the chyme. And just to put it into perspective with some FYI information here, you have daily 5 to 8 grams of sodium in your diet that you eat, but the amount that is secreted into the intestinal secretions is actually 25 to 35 grams. This is how much we actually need to be able to absorb all of that content of the chyme back. And of course, we need to absorb all of these electrolytes, the sodium back as well. So as I said, I will be talking about the secretion phase first. And what actually happens in the secretion, or how it starts, is with this small channel over here, which is the chloride channel. So all you need to know about it for now is that it needs cyclic AMP to function. So when there's cyclic AMP over here, this channel actually opens up and allows chloride to flow out of the cell. And if there is chloride flowing out of the cell, there must be chloride flowing into the cell from the other side, because how will you maintain the amount of chloride that is in the cell as well, right? So there's this channel over here, the potassium 2 chloride and sodium channel, that couples all of these ions together and puts it into the cell from the blood. So as I marked over here, this is the basolateral surface of the cell. Basolateral B, like it's from the blood, and the apical surface is facing towards the lumen. So I drew it here in orange. So as I was talking about this channel over here, it couples potassium 2 chloride and sodium, and this is how you actually get chloride into the cell, and you can have it flow out from this channel over here in the presence of cyclic AMP. Now, how does sodium flow out of the cell into the lumen? It's actually quite simple. It just follows the electrochemical gradient. So when chloride actually goes out, it creates this electronegative gradient over here in the lumen, and since sodium is positive, it has to follow it out. And as sodium and chloride are both over here, what follows the salt is actually water. So water has to follow it through the process of osmosis, and sodium can go out paracellularly, or there are other channels here for sodium that I didn't draw that also take out sodium. Now, hopefully that is clear, because now what I'm going to talk about is a pathology, which was talked about in the slides. And as you remember, this channel actually needs cyclic AMP to be open and for the chloride to flow out. Now imagine there was something that increased the concentration of cyclic AMP by a lot inside the cell, abnormally increased it a lot. This would actually cause this channel to be open excessively, and you'll have excessive amount of chloride flowing out, and consequently you'll have sodium following it and the water following it as well, and you'll be losing a lot of water in the through diarrhea. Okay, you'll be losing it into the intestinal lumen, and you'll not be able to absorb all of this water back. And this is exactly what happens when a toxin from the cholera virus actually affects the intestinal cells. So there's a receptor over here, this purple thing that I drew. It actually converts ATP into cyclic AMP. The cholera toxin actually binds to this receptor, activates it, and now you have automatically there's a lot of ATP converting into cyclic AMP. And when you have that, as we said, this channel opens up, this channel, and you have sodium and water following it, and you'll be losing a lot of water. So yeah. Now, actually, in this fluid that we said, so it has sodium, water, and chloride. Now, it actually has one more thing that I should have mentioned before, which is bicarbonate. So there is also bicarbonate flowing out into the lumen from the blood here. And Guyton and Hall actually mentions that the process that through which this happens is actually not very clear, but it should be, you should keep in mind that there is also bicarbonate flowing out, okay? Either through channels or through passively. You just have to know that it is flowing out, and which consequently makes the pH of the fluid that we're actually secreting into the lumen from around 7.5 to 8 pH, okay? So that's the pH of, it's slightly alkaline. Let's quickly conclude with the secretion phase over here. And we started with this chloride channel over here. And what it did was it pumped chloride out of the cell into the intestinal lumen. And we filled the cell back with this channel, which pumped two chlorides, a sodium and a potassium into the intestinal cell. And you also had channels that pumped out bicarbonate into the intestinal lumen. So all in all, you had chloride flowing out, you had sodium following it, and you also had bicarbonate. So we will see in the absorption phase that these are the three critical ions that we'll be dealing with, and we'll have to absorb them back. So let's go to it. The first scenario here is mostly focused on sodium, and the way you absorb sodium back from the lumen into the blood is first you create this gradient through this channel, this sodium-potassium channel. So as you know, it pumps out 3 sodiums and it pumps in 2 potassiums. And while doing so, what it does is it creates a gradient where there is high sodium here and there is very low sodium here. And you will have to replace the sodium that you just pumped out into the lumen, into the blood, by the apical surface. So you can take the sodium that is present in the lumen now, which you secreted before, and you can actually absorb it back with amino acids. So there's channels that carry sodium with amino acids, or channels that carry this sodium with glucose as well. And through the sodium, you'll be absorbing both the amino acids and the glucose. And this is how we actually absorb the content of the chyme and the nutrients from the chyme. So this is us dealing with the sodium. Now there are 2 other ions that we'll have to deal with, which is chloride and bicarbonate. Okay, so let's go to them. In order to talk about these 2 ions, they are dealt with differently in different parts of the intestine. So let's divide the intestine into 2 parts. A distal part, or the small intestine, into 2 parts, to be more specific. A distal small intestine and proximal small intestine. The proximal small intestine will consist of the duodenum and the jejunum, and the distal one will actually consist of the ileum, which I'll talk about later. So here we have the lumen, and here we have the blood. Now, as I said before, you will have sodium flowing in with sugar or amino acids, and you will have sugar and amino acids flowing out from the cell into the blood. And there are channels for that, and you can look into that from the lecture. And you also had this pump that I told you about, the sodium-potassium pump. So there now, what is happening is there is sodium flowing out, right? There is a lot of sodium in the blood, as I said, because of this gradient. And what will happen is that we also had a lot of chloride in the lumen that we secreted. So the chloride will actually follow this electrochemical gradient, and since there's a lot of positive charge here because of the sodium, the chloride will follow it, because chloride is negative and positive attracts negative. So this will just follow it. That's the way to remember it. You can just think there's a lot of sodium here, so what follows it is chloride. And that's how chloride is absorbed in the proximal part of the small intestine. Now, let's talk about the bicarbonate. Now, the scenario here is that in the duodenum, you just had pancreas dumping a lot of bicarbonate from the pancreatic juices into the lumen of the small intestine. So this currently the lumen has very high amount of bicarbonate, and you don't really need as much. So what you will have to do is you'll have to absorb them back into the cell. And the way you do that is you do that indirectly. There is this channel over here. It is a sodium hydrogen antiporter. So it pumps sodium into the cell and it pumps hydrogen out of the cell. And why this is quite good for us is because this hydrogen can actually go out, right, and combine with bicarbonate. Once it combines with bicarbonate through carbonic anhydrase enzyme, it creates carbon dioxide and water. This is actually very simple for us to absorb later on because, as I told you, there is a flow of ions, which is sodium and chloride here. What usually follows it is water, so you'll have the water flowing back into the blood. And also, by the way, carbon dioxide, it just diffuses passively into the blood and you respire it out through your lungs. Now, you can think of it that way, or you can also think of it this way. When you form carbon dioxide and water, what actually ends up happening is you absorb them back into the epithelial cell, and you do this passively, right? And these carbon dioxide and water through the carbonic anhydrase goes back and forms bicarbonate, and bicarbonate consequently leaves to the channel that it has over here on the basolateral side, so facing towards the blood. And that is how we deal with ions in the duodenum and the jejunum. Now you have a net absorption of two ions, which is sodium and bicarbonate. Okay, you also have chloride, but what they mostly focus on in this place of the intestine is sodium and bicarbonate, or sodium bicarbonate. Okay, now let's move on to the distal intestine, small intestine. Okay, distal small intestine, as I told you, was the ileum. Now in the ileum we deal with these ions differently. What happened before was we absorbed all of the bicarbonate, or most of the bicarbonate, back into the blood. We have deficiency of bicarbonate here. Let's call it that. Let's call it a deficiency of bicarbonate. But we actually need bicarbonate again in this place. Why? Because now there's a lot of bacteria that is present here towards, as the more distal you go in the intestine, you will find that there's more bacteria there. And going towards the large intestine and the distal small intestine, you'll find a lot more bacteria there. And what happens is these bacteria, they actually produce metabolic waste products that are acidic. Okay, so you need to neutralize these acidic byproducts through the use of bicarbonate. So you'll have to actually pump bicarbonate back into the lumen. And the way you do that now is a bit different, okay? You still have the normal channels that we had before, the sodium bringing in the sugar and the amino acid, and the sodium and the hydrogen antiporter, but now this channel over here is new, okay? So you have this channel that is an antiporter for bicarbonate and chloride. So it pumps bicarbonate into the lumen and it pumps chloride into the cell, the epithelial cell. And this is good for us because now we like in general we want to absorb chloride that we secreted in the intestinal crypts, and we want to pump out these bicarbonate because you need it. So that's how we deal with these ions on the luminal side, or the apical side of the epithelium. And going towards the basolateral side of the epithelium, or the one that's facing the blood, you actually have a channel for chloride here because you pumped it into the cell, if you remember. So it has to go out through the channel. If you remember, in the proximal part, or the duodenum and the jejunum, it was going out paracellularly. Here, since it's inside the cell, it will go out through these channels that are on the basolateral side of the epithelial cell. Now, if you want to conclude here, what you did was you absorbed sodium and you absorbed chloride, right? This was your net absorption, and you secreted bicarbonate. Okay, so that's how you dealt with the ions here in the distal part of the ileum. Okay, so that is it for the video. If you guys have a question, you can either text me or you can drop a comment and I'll reply to it as fast as I can. Okay, thank you so much, and Assalamu alaikum.